Good morning, everybody, and welcome to this week's installation of Micro Seminar. We hope everybody's doing well on this Friday, and hopefully, if you're in a place that is less restricted, have some good plans for the weekend. It's my pleasure this morning to introduce Aude Bernheim, who is at the Weizmann Institute currently. She did her PhD in microbiology at the Pasteur Institute, working on the evolution of CRISPR-Cas systems. She has a double master's degree and an undergraduate degree also from France, where she is originally from, and is moving to the French INSERM. I don't know if that's an institute or institute to set up her own shop. And so this is a really neat transition moment for her between postdocing and the next big step. And so we're very honored to have her presenting on a topic that we don't have a whole lot of experience with on Micro Seminar so far. So another great contribution. And with that, I'll let you take it away. Thank you.
So hi everyone, and first really thank you for allowing me to present this work. I'm super excited, and I will start dig in right away and start talking about my favorite subject, which is the conceptual, which is basically how bacteria and in more general prokaryotes defend themselves against viruses. So I don't see anything, so I hope you can see my screen and the presentation. So pretty much all cells, eukaryotic and prokaryotic, face the... Yeah, you don't see?
I don't see the screen yet.
Okay.
Sorry to interrupt you.
No, it's better than... And now, is it okay?
Yes, there you go, perfect.
Okay. So new start. So as I was saying, all cells, eukaryotic and prokaryotic, face the threat of viral infection. Here on the left, you have a bacteria that is being infected by viruses that we call bacteriophage or phage for short, but also plants or humans are infected by their own viruses. And all of these organisms, well, they have to find ways to defend themselves. And the way it goes is that when a virus comes, it tries to infect. And to prevent this from happening, well, cells will develop weapons or immune systems. But in response to this, the virus will then develop counter weapons that will render this immune system ineffective. And thus, the cell will have to find new ways to fight the virus and develop another type of weapon or immune system, and so on and so forth. And so there is a very strong evolutionary pressure for this immune system to diversify. And so the arms race that exists here drives the diversification of immune systems. And one of the consequences of this is that when you look at different organisms, and if you look, for example, at prokaryotes and eukaryotes, what you see is that you will have different types of immunity. The main immune systems, the main ways bacteria defend themselves against viruses is through restriction modification enzymes and CRISPR-Cas. But if you look for these in eukaryotic cells, you will not find it. Eukaryotic cells will defend themselves in other ways, such as NLR and RNA interference for plants, or for example, in the cell autonomous innate system of eukaryotes, through animals like humans, you will find interferon-stimulated genes and many antiviral effectors. And so this is the picture we've had for a very long time. But this picture has been starting to evolve. And one of the reasons why is that there has been quite a major change in how the field of what I call prokaryotic immunity, or how prokaryotes defend themselves against their viruses. For a very long time, we thought that bacteria mainly defend themselves through an innate immune system, mainly composed of restriction modification enzymes. And then in the year 2000s, the CRISPR-Cas system was discovered, and so pretty much prokaryotes got an adaptive immune system. There is also that we're known for specific example in specific bacteria, some abortive infection systems, which basically consist of when a pathogen comes in, it's recognized, and the cell will pretty much commit suicide which will prevent the fully infectious cycle of the virus to happen and thus prevent infection at the population level. But this is a picture we've had for quite a while now. But in the past few years, kind of the number of defense systems, of anti-phage defense system exploded. Chemical defense was discovered by Karen Maxwell and colleagues, signaling systems, and many other family of defense systems for which we don't know the mechanisms. And one of the reasons we kind of saw this explosion in the number of immune systems that are known in prokaryotes is because a strategy was developed to discover in a systematic manner microbial immune systems. And this all stemmed from an observation that was made by Kira Makarova and Eugene Koonin that showed something very peculiar about bacterial defense system is that they are clustered together on prokaryotic genomes. So here, what I'm showing is a line. Each line is a genome, and each of these rectangles is a gene. When the rectangles are the same colors, it means that this forms a system, a functional unit. And what you actually see here are all anti-phage defense systems, antiviral systems. So CRISPR-Cas systems, restriction modification. Brex is a form of, if we want, RM systems, also restriction modification. So what you see is that they are together in the same place. And this is what we call a defense island. But now, if we look at other types of defense island, we also see here CRISPR and restriction modification. But what we also see are these genes in gray. And gray in this picture means unknown function. And the hypothesis that was put forward by people in the lab that I'm currently working in, by the SOREC lab, and by Eugene Koonin and Kira Makarova, is that these defense islands actually have our defense systems, and that these genes in gray are novel type of defense systems. So this is what was hypothesized and tested in this study that was published in 2018. And indeed, when these systems were taken and put in bacteria that lack these defense systems, it was found that they are protective against phages. And so in this manner, more than 9 families of new defense systems were discovered. And I had the chance of pretty much joining the lab the day after this was published. And this is a strategy that you will see throughout this talk that we implemented to keep discovering, as the 10 defense systems that were discovered were kind of just the tip of the iceberg. And while regarding this discovery, one of the systems that was discovered in this study had tier domains proteins. And tier domains proteins are involved in both plant and mammalians immunity. And so with this discovery, it kind of showed that there was some conservation, as these tier domain proteins were also involved in prokaryotic immunity between prokaryotic and eukaryotic immunity. There were some conservation that actually exists. And later on, using the same type of strategy in our lab last year, it was also discovered that prokaryotes encode a pathway that is similar, that is pretty much homologous for some protein to the cGAS-STING pathway in humans. So there again, another proof that there are some conservation that exists despite this arms race. And another thing that came through other methods, not this defense island, but through other methods, was the discovery that prokaryotes also encodes prokaryotic argonauts, which are thought to be involved in prokaryotic immunity and that are very important in plant immunity. And so pretty much the question, which is pretty simple, are there more out there? Are these traces of conservation that started to emerge just something specific, or can we find more examples of this within the prokaryotic immune world? And this is pretty much what I'm going to discuss today. And I will talk to you about 2 stories, one in detail and one more briefly, which I hope will illustrate how much this comparative immunology approach, by getting inspired by other type immunology, can open new leads in prokaryotic immunology. And so the first story I want to share with you is about something called viperins. And hopefully you'll get to read about it soon, and it's currently under review. So what is viperin? Viperin is an antiviral gene that was discovered in humans. Pretty much, when an RNA virus comes in, it is sensed by the cell, and the cell responds by expressing viperin. And this viperin elicits an antiviral activity. So while this one was known for quite some years, the mechanism was only discovered in June 2018, so 2 years ago. And Gizi and colleagues actually discovered that viperin is an enzyme that catalyzes the transformation of CTP, so the RNA nucleotide we all know and love, to a modified nucleotide called DDH-CTP. And the modification is happening here in red, mostly by removing this hydroxyl group, this OH, and creates a double bond. And why is this important? Well, if you incorporate this nucleotide into your RNA, because it's missing this OH group, you cannot, after that, incorporate another nucleotide. The chain is what we call terminated. And so this molecule is called an RNA chain terminated. And so the mechanism that pretty much Gizi and colleagues discovered is that what happens is that when the virus comes in, the viperin takes CTP, makes this molecule, DDH-CTP, that is incorporated by the viral polymerase in the RNA transcript, but not by the polymerase of the host. And this leads to a selective antiviral activity. The viperin is pretty much poisoning the virus without poisoning the host. And when this paper came out, we were super excited because it was the first time that this type of immune strategy was described, the natural production of an RNA chain terminator. And we were wondering, well, maybe, of chance, these exist also in prokaryotes. And you already guessed the following, that indeed, we found prokaryotic homologs of the viperins. What I'm showing you here, each line again is a different genome. Each of these rectangles is a gene. And you have here in red homologs of the human viperin. It was very impressive. Some of the homologs had up to 37% homology, so very high homology. And we directly went and looked at what was around those prokaryotic viperins. And what we found is that there were defense systems. So pretty much we could say that the homologs of the human viperins in prokaryotes, they sit next to defense systems. And for us, it's a very good predictor of the antiviral function of potentially this protein. So this suggests that the genetic context suggests antiviral activity. And in that case, it would mean that the prokaryotic viperins and the human viperins would have the same function. And so we went on and tested for that. So the way we did it is by taking examples, candidate genes from diverse bacteria. They were not in regular, let's say, E. coli, bacillus, something that it's easy to work with. So we just took the DNA sequence and expressed it in an heterologous manner in E. coli, and then tested if we could observe antiviral activity. So the way we do that is that here in the y-axis, you have the Aude 600, and in the x-axis, the time. So we're following the growth of the bacteria. Here, this is a negative control. We see the bacteria growing. When we add a phage, in this case, phage T7, what we observe is that the population collapses, which shows that it died because of the phage infection. Now, when we go the strain, E. coli strain that heterologously express the pVip, pVip stands for prokaryotic viperin, that's how the little name we gave them. We see that it grows, but now if we add a phage, what happened? Well, pretty much nothing. The population is able to grow despite the presence of the phage, which means that this prokaryotic viperin or pVip brings antiviral activity. And for us, we were very happy about this because this was showing that indeed, both in prokaryotes and eukaryotes, the viperins have the same activity. But we are wondering how diverse, how many candidates, how widespread was it? So what we did was to look for homologs of these pVips in around 60,000 genomes, and we found 218 that were non-redundant, which means that it's a rare protein. It's not very common at all. But one surprising aspect is that when we built a phylogenetic tree to see how the family organized and looked from where the different pVips came, we saw that they were coming from diverse phyla. They were not restricted to one specific species, for example. They were present in Archaea, cyanobacteria, proteobacteria, firmicutes, etc. And for us, this kind of pointed out to probably a very ancient origin of this protein. And at this point, we're very curious of seeing where would the eukaryotic viperins actually form this tree. And what we saw is that they form here a monophyletic clade, so they are all together, and they all stem from pretty much this prokaryotic viperin family, from the tree. So we see that pretty much there is a prokaryotic origin of the eukaryotic viperins. And another feature that stems directly is that if you think that this is all the eukaryotes we looked at, and those goes from unicellular to multicellular, really try to sample the diversity, we see that it only represents a small part of the diversity that is present in the prokaryotic world for this family of proteins. And at this point, we thought, well, maybe this diversity is also representative of diverse function. Maybe not all prokaryotic viperins have the same function because they are more diverse. So we wanted to be sure or not that all pVips are antiviral. And so to do this, we took pVips from all over this tree, tried to sample the diversity on the tree, and tested them in the manner that I just showed you. And what we were able to show is that 28, so around half of them, show antiphage activity. And what does this mean? That first, it's really pieces of DNA that come from very, very diverse bugs, Archaea, cyanobacteria, proteobacteria. You put them into E. coli, and they provide antiphage activity. So this kind of points out to a conserved mechanism. Another aspect is that we had candidates that provided antiphage activity from all over the tree, which pretty much convinced us that indeed the whole family was antiviral. And following the same approach, we were curious to see if, by using the same strategy, what would happen with the human viperin. And with this strategy, we were able to show that the human viperin, so these human pieces of DNA, provides defense against prokaryotic viruses. So pretty much, we show that here we have all this family that seems to somehow be able to provide defense against phages in E. coli. So at this point, we had the diversity and the conservation. And we know that in humans, the human viperin produces a specific molecule called DDH nucleotides. So what we wondered is that, are all of these pVips producing such type of molecule? Are they producing all type of molecule? So we wanted to know more about kind of the biochemistry behind it. And more specifically, do they generate different molecules? So the first clue to answer this question came from the inspection of the genomic context of the human viperin. Gizi and colleagues showed that right next to the human viperin, there is a citidylate kinase called CMPK2, and that it's important in the antiviral function of the human viperin. What it does is that it takes CMP, so the monophosphate version of the molecule, adds phosphates, and pretty much you get CTP. And then CTP is the substrate of the human viperin that makes DDH CTP. And when we now looked at our own pVips, we did see that in 20% of the cases, our pVips also had neighboring kinases. And in some cases, similar to the human, the kinases were citidylate. But in other cases, the kinases were timidylate or adenylate. And this kind of pointed to us that maybe if this is the reaction that is happening with the human viperin, CMP to CTP to DDH CTP, well, this is the reaction that might happen in prokaryotes, GMP to GTP to DDH GTP, or UMP to UTP to DDH UTP. And this was for us kind of a clue that we might look at different types of products of the potential pVips products. So to be sure, we set out an experimental system. So what we did is to grow our bacteria that we induced. We took the pellet, so only the cells, and extracted from the cells the small molecule fraction that is there, and then sent it for analysis to do untargeted metabolomics. And what we observe, I'm going to walk you through the graphs, is that the way you're looking at this, we have here a negative control. And then because we know what the human viperin is producing, we have a positive control. And for each of these graphs, the way you look at it is that you're plotting the intensity for a very specific match detected by LCMS. And for each product, for each molecule, the presence of this molecule will be by the presence of a peak at a specific retention time at this mass we extracted. And so in a negative control, it will look like this, nothing. And here, it means that we do see the presence of this molecule DDH-CTP in the human viperin sample. And for us, this was really something we're extremely happy about, because it meant that with this technique, with these methods, we were able to detect this DDH-CTP in bacteria cell lysates. But the DDH-CTP is not the only thing we saw. We also saw presence in the human viperin sample of what we call DDH-CTP derivatives, so DDHC, the same molecule without the phosphates, or DDH-CMP. And the way we interpreted it is that it's not really stable, maybe the triphosphate version in the type of buffer and experimental condition we used. But this is a signature of the type of molecule that is produced by human viperin, in this case, DDH-CTP. So, of course, what's next? We wanted to check if we could detect these molecules in our own pVips. So this is what we did. Here, I'm showing you only a summary of our results based on the graphs that I just presented to you. And so pretty much, human viperin, it has DDH-CTP. And when we look for DDH-CTP in our pVips, we did find some that also produces DDH-CTP, pVip 6, pVip 50. But for others, we didn't find traces of DDH-CTP. However, because we had the idea with the kinases that maybe other products were involved, well, we looked for others. And what did we find? We did find pVips that produce DDH-UTP derivatives, and pVips that produce DDH-GTP derivatives. And these are new types of molecules that were not observed up to now in nature. And another very exciting aspect is that when we looked further, some of our pVips did not produce one product. They produced several products. They were able to catalyze both CTP to DDH-CTP and UTP to DDH-UTP. pVip 58 was able to pretty much take in all the three of them. And this means that pretty much these enzymes are promiscuous. They are able to take a nucleotide, either one of the three or two out of the three, and do this modification that changes a nucleotide into a poison for a virus, these DDH nucleotides. And so for us, this result allowed us to conclude that prokaryotic viperin produce diverse antiviral molecules. One side note that I find quite interesting is that in 5 of our pVips, we are not able to detect any of these molecules. So maybe, you don't see DDH-ATP here. Maybe it's DDH-ATP and we don't have the mean or the technical to observe these molecules. Or maybe they also produce something else that we don't know about and that this technique was not able to detect. But for sure, this is a very exciting thing to go on and try to find what these are producing. Because these, just to be clear, do elicit a very strong antiviral activity. So at this stage, we now know that those prokaryotic viperins produce diverse molecules. The question remains, how exactly do they work? How do pVips protect bacteria against phages? And so then again, we got inspired by what happens in humans. So in humans, the DDH-CTP molecule is incorporated by the polymerase of the virus in the RNA transcripts. And then it leads to the antiviral activity. So we decided to go for the same hypothesis and to check if this was also the case for our pVips and also for the ones that express, that produce these new types of molecules. We devised a reporter system. And the reporter system works as follows. We have a GFP reporter gene that is under the control of a promoter, of a viral promoter, meaning that the transcription of the GFP depends on the T7 RNA polymerase, which is a viral polymerase. So what happens that when we express the viral polymerase, It comes and then it expresses a GFP. We get GFP transcripts and a functional GFP signal. However, if now we add the pVip in the system by inducing the pVip will produce DDH nucleotide. And what we expect to happen is that for the GFP gene, that is a single gene under the control of the viral polymerase, pretty much the DDH nucleotide will be incorporated by the viral polymerase. And so the transcripts will not terminate properly, and we will not see a functional GFP signal. And these are the results. So here in the y-axis, you see the fluorescence over OD, over on the x-axis the time. In gray, when we don't induce GFP at all. In green, when we induce GFP. And in red, when we induce GFP and the pVip. This is a negative control. There is no real difference between the GFP and the GFP and pVip induced. However, when we did this experiment with the human viperin, you can see that the GFP signal is completely abolished when we express the human viperin. And this was also the case for other of our pVips like pVip8 and pVip9. And just to be sure that this was happening at the RNA level, what we did was to take RNA at this point and to see with RNA-seq what the transcription looks like. And this is the result of the percentage of GFP expression compared to the negative control. And you basically see that all the GFP expression is greatly reduced when we express any of these pVips, the human viperin, while at the same time not showing it here, but for a gene under a bacterial promoter, we don't see such reduction at all. This is specific to the viral polymerase. And so this led us to conclude on this model that if this is what's happening on the left in the human cells, maybe this is what is happening in a bacterial cell. You have diverse nucleotides for pVip that will modify several, or one single that will be modified by the pVip. And the pVip will generate several nucleotides that will pretty much be incorporated by the RNA polymerase, and it will lead to a premature termination of the phage transcript and an antiviral activity. And it's very, I think, fascinating how much the strategies and here the sequences and the way all of this is conserved between eukaryotes and prokaryotes. But the strategy of chain on RNA chain termination and chain termination in general, antiviral strategy, is not only shared by eukaryotes and prokaryotes, it's also something that before it was discovered in nature was used as a therapeutic strategy. This molecule is CTP, the nucleotide CTP. And for the DDH CTP, it's removing here. But if now you don't remove the OH, but you change it here to this group, what you get is AZT, which is an antiviral drug against HIV. And actually, other and many antiviral drugs are terminators, such as acyclovir or telbivudine. And so what we think we might have with all of these pVips that produce these diverse antiviral molecules is maybe potentially new antiviral drugs. We haven't tested it yet, but maybe why not? And so just to wrap up kind of this story of the viperins, what we have found is this antiviral strategy that is conserved between prokaryotes and eukaryotes, but with somehow a much bigger diversity in the prokaryotic world. It's a bit, according to me, as many things like for the people who know the CRISPR, where you have one concept of making cuts in DNA, but in a very different manner at the molecular levels. Here again, we have one variant that exists and is very interesting in humans. And because it exists in the prokaryotic world, we have much more diverse variants. And one aspect that I find particularly exciting is that chemical defense was also described. This is what we call chemical defense, meaning that you create a molecule that is toxic for the virus, but not for the host. And this was also described in Streptomyces species by Caron Maxwell and colleagues. So this is a second example of chemical defense. And if we now know that bacteria creates and produces these small molecules that are toxic for viruses and maybe for human viruses, then maybe the same way that bacteria has been used for years as repository for antibiotics, here, by going after new type of chemical defense, new type of strategy of how bacteria produce these molecules, maybe there is a way to find new antiviral molecules and maybe drugs. So this is the first story, I think, really shows how everything is kind of conserved. It's fantastic to see the level of conservation that exists between prokaryotic and eukaryotic immunity on this question. And by eukaryotes here, it's humans. The second story that I want to tell you about is in less details, is with an insight from plants. And as you will see, it's much more, I would say, in terms of inspiration and conceptual inspiration than actual conservation at the sequence level, for example. And this story is about retrons, and I will get in a minute to what retrons are. But first, I want to say that this was the result of a collaboration with two amazing scientists, Adi Milman and Abigail Stockar-Avichai, who I really had the chance to work with. And I'm not going to go into deep experimental details about this story, but you can find all the details in our preprint on bioRxiv. So please do go check it. And now let's go for it. So what is a retron? So retrons are elements that were discovered in the 80s because these elements produce a satellite DNA that were visible on gels. And so people saw this presence of this small DNA, and so they went on and tried to understand how was it produced. And what they discovered is these elements, retrons, that are described with two parts. You have on one aspect a non-coding RNA, and then a reverse transcriptase. And when these are expressed, the non-coding RNA will fold in a very specific way with a very specific structure, and the reverse transcriptase will start reverse transcribing from this specific guanosine. And so it starts reverse transcribing while the RNase H is degrading the RNA part, and in the end, you end up with an RNA-DNA hybrid, which is called msDNA, and which is what you see on the gel. And so several retrons, 16, have been described experimentally, characterized, and they have also been used as tools in synthetic biology and genome engineering. And you can find a great review about this on this subject that I'm just showing an illustration about. But despite all this intensive biochemical work, their physiological function has remained elusive. No one knew what they were doing. 30 years, no clue. There were some hypotheses, but not definitive proof. And this is pretty much where we come in. And as many that today, we were basically looking at defense islands. We found the systems. It was defensive. And how do you know it's defensive? Well, here, these are phages that I'm showing, each time tenfold dilutions. And here, pretty much, this means that the phage are able to infect. What you see here in the background is the bacterial load. So when you see a hole, it means the phage kills the bacteria, no more bacteria, the phage is able to replicate. However, when you have a defense systems, what you see here is that it's much more faded, and here the phage is not able to replicate at this concentration on the bacterial load. This is a defense phenotype. So we have this defense system. And when we look at this defense system, it had mainly three parts: a gene with an ATPase domain, an intergenic region, and a reverse transcriptase. And when we looked a bit more into it and did a couple experiments, what we found out is that this part is actually a retron, which means that pretty much here, there is this non-coding RNA with the retron features, and we're able to show that this retron produces this small msDNA that you can see on the gel. And this means that you have a novel defense system that encodes a retron. And the retron properties are essential for the defense. So we thought maybe all retrons are actually involved in antiviral activity. Maybe this is their function. And one key aspect, I think, of the work was that now I described to you what is a retron. It's a reverse transcriptase and a non-coding RNA. However, in the system that I just showed you, our novel defense system, what we had seen is that actually there was another part, another gene, the second gene that was involved in the defense. And the second gene was also essential for the defense phenotype. So here, what I'm listing is the known retrons that were experimentally validated. And we went and looked a bit more in detail and tried to see if maybe all of these retrons also came with cognate gene, associated genes. And indeed, all of these retrons came within systems. So they had associated genes that we believe are essential for the defense. So what we did was to take all these known validated characterized retrons, but took them not only the non-coding and the reverse transcriptase, the non-coding RNA and the reverse transcriptase, but also the associated genes. And we put them in E. coli and tested them against diverse phages. We tested all of them except these ones that came from Myxococcus. And for all of these, we had at least one example of a system that provided defense against phages. So all of these retrons, we were able to see that they were protecting against phages and against diverse phages. So for us, this was a proof that this was a physiological function of retrons, and that pretty much we had the answer of a 30-year-old question, which we were super happy about. But of course, we wanted to know how they work. So, and this is when the insight from plant immunity really came to help us and helped us crack the mechanism. So in plant immunity, you have something that was going to be termed the guard hypothesis. The way it works is that you have a host protein, and when a pathogen, typically a virus, can come, it will modify the host protein in some way, and this modification will trigger an immune response. Why? Because there is a protein that is involved, an immune protein that guards this host protein and will sense the modification of the host protein. So when the host protein is modified, it will trigger the immune response. And I will skip all the experimental work that led to decipher the molecular mechanisms, but for a very specific retron called EC48, this is the proposed mechanism we ended up with. And it is that EC48 guards RegBCD. The RegBCD complex is a complex involved in DNA repair, but that is also very important for immunity. The reason is that it's an exonuclease that when a phage comes in, it degrades its DNA. So what do phages do? They encode RegBCD inhibitor. And what we found out in our experiment is that pretty much when you have this RegBCD inhibitor, they will inhibit RegBCD, and this is sensed by the retrons that will then activate the effector and leads to cell death. And cell death is what we call an abortive infection phenotype, meaning that it will prevent the virus to complete its infectious cycle, and thus the virus will not be able to replicate and to infect the whole population. And so just a quick summary on this. Pretty much we managed through this discovery, through the defense island, and the discovery that retron comes within the system, that retron's physiological function is antiviral, and it's actually a widespread and diverse new type of antiviral system in bacteria. And we also discovered by looking at the mechanism of EC48 that the guardian hypothesis also works in bacteria. And why is this also very important? We now have experimental ways to test for this type of hypothesis, which protein is guarded by which system and how. So here I'm showing you one example, but maybe other retrons guard other types of proteins, or maybe other defense systems of which we don't know the mechanisms, they also guard our guardian systems. And with this kind of new conceptual lead, we can now go in and really interrogate in a novel way, with very simple experimental setup, how if this is truly the case. And I still find it quite fascinating that here we have no conservation in sequences, no the same, everything is different, but the concept remains. The fact that you pretty much converge to understand how you have been infected, and that this will trigger the immune response, that you need to guard a very important host protein, this has been conserved between at least plant and bacteria. And so I just want to wrap up this seminar with a few ideas on what I like to call the power of comparative immunology, that today was about new prokaryotic defense systems. However, this conservation can go in another way. We have systems that are conserved between humans and plants and bacteria. And in bacteria, we start knowing a lot of different defense systems, anti-phage defense systems. So maybe in organisms that do face viral infection and for which it's very important, I'm thinking here, for example, with Emiliana Huxley, maybe you can get inspired by finding immune systems in these organisms that are actually from bacteria. It's a way to start. A second aspect is that that was exemplified with the retron stories, that we can get inspired by things that were already known in eukaryotic immunity and start applying them to prokaryotic immunity to discover new mechanisms or new ways of thinking. Well, maybe other types of concepts that are very big in eukaryotic immunity exist in prokaryotes. You know, interferon, that is a molecule that report the fact that a cell has been infected to other cells. Well, maybe this also exists in prokaryotes. I don't know, maybe. The other side of the coin on this conservation aspect is that if we now think that there are systems like the viperin systems that are very similar between eukaryotes and prokaryotes, there are fundamental questions regarding these systems that could be answered in studying them in bacteria. Typically, the cell versus non-self discrimination in this system, meaning how is the host able to discriminate and say, no, I don't want this nucleotide, while the viral polymerase takes it, are things that could be studied in a bacteria setup. And this could give insights then to immune mechanisms in eukaryotes. And what I want to leave on the note I want to leave on is about prokaryotic diversity. As I told you, for the viperin, it was amazing to see that when you have something that is very interesting present in one version in humans, well, in prokaryotes, we have it present in many, many versions. But even more exciting, with all the story I've been telling you, the diversity of immune systems that exist really, for me, is a sign that many, many other types of systems exist. I'm convinced other type of chemical defense system exist, but maybe other things that are extremely exciting. And I think we're only at the beginning of discovering how bacteria defend themselves against viruses, and that there is much more to do there. And on this, I, of course, want to thank everyone that has been in this work. First and foremost, my postdoc mentor, Rotem Sorek, so it's his lab, and who was absolutely a fantastic mentor and really helped me and supported me in all of the science that I have been doing there, and for building the most fantastic teams that existed in science. So the Sorek lab is really a fantastic team. So for the retrons, again, I want to acknowledge Adi and Abigail, who really did all the work with me. And for the viperins, again, Adi Milman, who did a lot of bioinformatics with me, and Gal and Gil for conceptual aspect, and Gilad, Carmel, and Sarah for helping me in doing some of the experiments, and our collaborators in company. And as it was mentioned recently, I'm moving from Israel in Weizmann to Paris. So if you are in Paris in this time and want to say hi, please, you can contact me. And I want to thank you all for listening to me.
Thank you. That was a wonderful talk. Absolutely fascinating. I'm continually impressed by the new findings in immune systems of prokaryotes, and this was a wonderful addition. We have several questions from the audience. So let me start at the top from Urine. Hello, did you express the different pVips you found by bioinformatics in E. coli? And you also expressed the related kinases. Did you also express the related kinases?
So we only we only expressed the pVips. We thought maybe if it doesn't work with just the pVips, we would also express a related kinase. However, it worked, and somehow I think it's because you have enough nucleotides there to be the substrate. A second aspect is that one thing that I didn't mention is that some of the pVips are actually fused to the kinase, And we had one example in our set of homologs we tested that had the fused kinase. And we didn't notice anything specifically different. It had antiviral activity, but not really more. So also only 20% of our pVips actually had neighboring kinase. So a lot of the homologs we tested didn't even in the wild seem to have kinases nearby that would help them to perform their antiviral activity.
Great. Okay, from Sam Sternberg. Hi, Aude. Have you tested phage versus host RNAP specific inhibition in vitro? Is it surprising that these chain terminators are so polymerase specific? And are viperins conferring actual resistance then, not ABI?
So for the in vitro part, we actually did not perform this experiment, but it was performed by Gizi and colleague in the previous paper. And they had noticed that indeed it was polymerase specific. You had different results with different polymerase. So this has already kind of been established by them that the tolerance to DDH-CTP diverge in terms of polymerase in vitro. And the second part of the question was?
Are viperins conferring actual resistance then, not ABI?
Yes, ABI. So yes, viperins, sometimes the resistance aspect, you have two types of phenotypes. Either it delays the death, or it really allows the viperin to completely survive. So we didn't see ABI type of phenotypes.
Great. From Cian Owen, amazing talk. The RNAP specificity is incredible. How do you think this is achieved by such tiny proteins? And are phages that use host RNAP intrinsically resistant?
So that's a question we've been asking ourselves quite a lot. So one hypothesis we have, I don't have any proof. The phage I've been talking about mostly is a phage called T7. And T7 completes its cycle in 17 minutes. So it's extremely fast phage. So we think that maybe because the virus are so much faster, they cannot do more proofreading. And maybe there is a proofreading mechanism that is present within the host type of polymerase that indeed somehow allows this self versus non-self discrimination. But I don't have proof. The second about phages that uses the host polymerase, we actually have evidence that this prokaryotic viperin protects against phage lambda. And phage lambda is known to use the RNA polymerase of the host. So this was quite a riddle. So there are a few possibilities there. One thing is that in the lambda life cycle of the phage, one of the first things that is expressed is something that will then kind of modify the RNA polymerase of the host so that it's favoring the genes of lambda. So maybe this type of modification could make them sensitive. Or maybe there is something else happening. Maybe while the pVips is modifying some nucleotides, maybe a subproduct is produced. Or maybe the product then is degraded into something else. Or maybe there is a nucleotide specific produced by the phage that will be somehow catalyzed by the viperin. So we don't have a clear answer regarding this, but for sure it's something that kind of drives us to think, is it the specificities that change, or are there additional mechanisms that provide antiviral activity for this phage? This we don't know yet.
Great. Another question from Eurene. Why do you think viperins are not so widespread in prokaryotes?
So who knows? It's very interesting, the distribution, because if you look at usually the distribution of defense systems, it really looks like a lot of horizontal gene transfer. And here doesn't seem to be the case. So I absolutely don't have a good explanation for this distribution. Maybe it's not that effective. Maybe it's costly to produce these DDH nucleotides. Maybe there are many things. One of the issues we currently have is that they're not present in model organisms. So we are only studying them through the overexpression in an heterologous bacteria. So maybe there are some costs that are inherent to the systems that we cannot see because we cannot for now study them in a natural context.
Yeah, that was a really,
to add one thing, for me, my hypothesis is that they have been existing for a long time, and pretty much they've been lost in a lot of cases. And what we see are trace. Another hypothesis I forgot to mention regarding this is that for now we've looked at pVips in a very conservative manner, meaning that we have the sequences are quite closely related. And one thing that is possible is that this form of anti-phage defense is actually still present, but in a sequence manner that is the sequence evolved, and so we don't see them as part of this nice phylogenetic tree family, but that they still exist in other forms and still produce maybe the same molecule or others, but we cannot find them through our homology search approach that is based on sequence homology.
Yeah, I think that's a really great possible answer. Yeah, it's a fascinating idea that these things are distributed in a sort of, you know, inconsistent way. But I think yeah, I think searching for more divergent sequences would be a really great avenue to take. Another couple questions from a handle named HojoMan85. What is the significance of retron nucleic acid being composed of an RNA DNA hybrid to the guard function?
Wow, amazing. So on this, good question. I remember being in a conference and someone saying like, don't, we were with the Adi and Abigail, the other collaborator, and we're like, don't focus on this weird DNA RNA thing, because of course, why would you need the DNA and RNA? It's very weird. Why such a complicated thing? And I would really encourage you, other preprints were just posted on retrons by the NASA STIPAS group, where they start answering this. And the answer could be that by having an RNA and DNA part, you can maybe sense, maybe not in the guard hypothesis, maybe in another type of retron hypothesis, you can sense diverse cues. You know, maybe the phage has an RNase activity that could degrade the RNA, and then it would trigger the system. And then maybe it can sense something through the DNA part. So maybe the fact of having these both like RNA and DNA can be just to sense in different manners that the cell has been infected. But the key thing that we don't know yet, but we're starting to think that the retron part is a specificity part, and somehow is able to sense the phage infection, or through the guard hypothesis to sense that it has been modified. And maybe the fact that it has a protein, an RNA, and a DNA part makes it kind of many possibility to sense many cues of infection.
Okay, so another question, or part of the same question from the same person. What kind of functional information do you think could be inferred from retron sequences?
So then again, I think this is a mystery to discover. For now, what is striking is that the retron sequences of the non-coding RNA are very different. What seems more conserved is its specific structures. However, you need specific sequence. You can kind of mix and match the reverse transcriptase and the non-coding RNA. And when you don't have too much difference, then the reverse transcriptase can still manage to reverse transcribe. But when you have too much difference, it doesn't work. So it seems that there is really a co-evolution between the reverse transcriptase and the non-coding RNA. For now, as we don't really know what this non-coding RNA, this msDNA, RNA DNA part does, it's hard to say what the sequence actually means. We try to look for clues if it looks like phage DNA or something. We didn't find for now any good avenues that would give us idea of something that is hidden in this sequence. Contrary, for example, to CRISPR that had the repeats and the spacers that match the phage. So for now, we haven't found anything of this sort.
Interesting. So yeah, I have a couple questions. That's all the questions we have for the audience right now. But because we're on a lag, I'm going to let the information flow over there and see if we get any more. But I have a couple questions in the meantime. You know, I'm thinking about all these different immune systems and several that you were discussing today. So that you got the viperins, and then you've got the retrons, but of course there's a CRISPR-Cas too. Some organisms have one of these, some organisms have two, some organisms have all three, some have none. So I'm wondering, you know, what's going on with the proliferation of defense systems, and what does that tell us about the lifestyles of these organisms, and why might you need lots of different defense systems versus one, or, you know, why would an organism potentially hold on to one preferentially? Anyway, I don't know if you could just get into sort of what are the implications of this multifaceted but unevenly distributed collection of systems.
So one thing, I will give a few elements on that, but if you want to really know more, we wrote a perspective piece on that in Nature Microbiology, Reviews Microbiology, that's called the pan genome immune system, something like that, pan immune systems of bacteria. So here we discuss exactly this. Why would you need several defense systems? I showed you defense items, and you have like, you say 3, but can go to 10% of the genome that is dedicated to defense. And what we think is that first, defense systems, they only protect against a subset of phages. So we never found a defense system that is able to defend against all phages. And the reason why is that some phages won't trigger the defense system. But another reason is that during the arms race that I was discussing, the phages will develop counter weapons. So for example, if you have a CRISPR, phages will have anti-CRISPR. And so if you then want to answer, you can develop anti-CRISPR, or you can also develop another type of CRISPR, or just get a new type of defense systems that will kind of bypass the whole thing. And we think that these two things, that not all defense systems can not, there is not a single defense system that can protect you against all phages, and the fact that the phages have like anti-immune systems really leads to the diversification. Then you also have these defense systems that are super mobile. We find them like really always exchange and things like that. So this is kind of another thing that I think is kind of linked, that you need to exchange them all the time in order to be able to have the one that will kind of work out for the phage that doesn't have the counter weapon. And in terms of lifestyle, my view is that for those who have zeros, either they are symbionts and like live inside other bacteria, else I believe it's just we haven't found them for now. That's my view on things. Maybe you have some that will have less because they will face like less diverse types of phages. This is a possibility. It remains to be kind of demonstrated. But I think for now, a lot of the things we just didn't know. And so we couldn't find them.
Great. Yeah, it's a really exciting future moving forward for some of these. You know, a lot of the texts I study don't have canonical defenses, at least of the types that we've looked for so far. But this possibility that there's plenty of them, that they're right under our nose and we just haven't figured out what they are yet, is really exciting. So I hope you're right. That sounds awesome.
That's the optimistic view.
Okay, well, so that's all the questions we have so far, but people can get in touch with you, obviously, if they have others. And I just wanted to thank you again for a really fascinating talk, and thank you for your contribution to the micro seminar.
Thank you.
Okay, bye-bye. Okay, Aude, if you're still there.
I'm still there.